Hi, I would like to welcome you all to this webinar about the MoTrPAC Data Hub and the data that you can access there. So MoTrPAC is short for Molecular Transducers of Physical Activity Consortium, which is an NIH Common Fund program that aims to build a map of the molecular responses to exercise. My name is Malene Lindholm. I represent the Bioinformatics Center in MoTrPAC, led by Dr. Ashley and Dr. Wheeler at Stanford University. And I will be one of several MoTrPAC members that you will hear from today. So I will start by giving you a brief introduction to MoTrPAC. Then I will hand over to a representative from the preclinical animal study sites that will walk you through the exercise intervention protocol and tissue sampling in the preclinical arm of MoTrPAC. Then we will get a brief introduction to the different molecular assays that have been run on the animal tissues by representatives from the chemical analysis sites in MoTrPAC. And after that, we will describe the different types of data on the Data Hub, and importantly, how you can access these data for your own analyses. After the presentation, there will be time for questions through the hand raise function and the Q&A tab. During the presentation, you can also ask questions through the Q&A function, and we have multiple MoTrPAC members who will do their best to answer them. So you can see the Q&A tab at the bottom. So please test it out and ask whatever questions you may have. So what is MoTrPAC? So as I mentioned, it's short for the Molecular Transducers of Physical Activity Consortium. It's a large NIH Common Fund program that spans the mainland US. There are 19 awards in total, distributed across many institutions. So there are 7 main clinical centers, 3 main preclinical animal study sites, 7 main chemical analysis sites, a consortium coordinating center that is spread across 3 different sites, and a bioinformatics center. And MoTrPAC is an ongoing study. There is a human clinical study arm in progress that includes endurance and resistance exercise. But for this webinar, we will focus on the preclinical arm of MoTrPAC. And for that part, I will hand over to Dr. Karen Esser, professor of physiology at the University of Florida, who is one of the principal investigators for the animal intervention in MoTrPAC.
Okay, thanks, Malene. So you can go ahead and move to the next slide. So for today's webinar, we've divided what I'm going to present to you into 2 sections. The first section is really describing the protocol and the tissues that are comprising the available data that has been released publicly. So that will define that. The second part of this presentation is the rest of the animal studies as well as all the tissue archives. And I have also included a link on this website because we have I won't have time to get into a lot of the details of the animal protocol. If you're interested in sort of more of the details of the animal protocol, please feel free to download the protocol that's on the website, and that will give you a lot more information. Next. Okay. So to start with, so there's a certain amount of data that is currently publicly available. And this comes from studies that were using Fisher 344 male and female rats. And these rats came out of the NIH colony. We used treadmill exercise, and these tissues come from the acute response to exercise time course. The age of the rats is 6 months of age. And just a general feature of this is that these animals were under 12-hour dark and 12-hour light cycle. We reversed it, so all of our exercise was occurring during the early part of the dark phase or the active phase for the animal. And this short table over on the right just gives you a listing of the tissues that were used for all the chemical analysis sites. So blood, gastrocnemius muscle, heart, liver, and white adipose tissue. And the data from that it has been released and is available to you right now. Okay, next slide. So the protocol for this acute response is illustrated in the cartoon at the bottom. But basically, some of the, as I said, some of the main points here is that we removed, and we just, one thing I do want to make clear is this protocol came from all three PIs for the preclinical sites coming together, but also in a lot of consultation with the human exercise. So we can actually try, we're trying to, you know, do some things as much as we can in parallel. So in this case, we remove food 1 hour before lights off. And just as a reminder, since rats are nocturnal, they're not eating a whole lot overnight. So it's somewhat similar to an overnight fast prior to exercise. We have a little bit of a unique situation for the 7-hour exercise and the 7-hour control groups. Rats were provided with 1 to 2 food pellets for only 2 hours at the sort of immediate post-exercise time point. So we did our best to try to control for feeding with such a large study. The bout of exercise was 30 minutes of treadmill running. At the end of the day, we are over 70% VO2 max, but we were trying to be close to and just above. And the speed and slope for that, for the either female or male rats, is given below. All the tissues were collected at 7 different post-exercise time points, and those are listed in the line graph below. And then some data that isn't normally available with these kinds of studies is we actually kept the time from the death of the animal to the time that the tissue was frozen, and this is all recorded and available. The tissues were flash frozen and shipped to the central site, which is held at University of Vermont, where they were either processed for chemical analysis or they're stored as archival. And then we did have 2 time point controls. All right, so now very quickly, I'm gonna move over to the rest of the studies. So again, there are 3 preclinical animal sites where, and across these sites, we really worked quite hard to standardize our lighting, our feed, or where we got our chow, and the bedding, and we stayed within what the NIA colony is maintained on. Same Fisher 344 rats, treadmill exercise. So in addition to our acute response to exercise, we're now going to be, we also have exercise training, and we're going to collect some samples at different times during the training adaptations. We had 2 study ages. So there were the 6-month adult animals and then late middle-aged or 18-month-old Fisher 344s. Same light cycle. And then just as a highlight here, we, you know, one of the strengths of the past is that we collected approximately 19 tissues for each rat. And just to give you a sense of all the tissues that are stored, that's on the table there on the right side. So different parts of the central nervous system, different organ systems, and obviously things that have never been studied before with exercise. Okay. So I'm not going to spend much time on this because this is the same, virtually the same sort of cartoon diagram, but we had the acute responses were performed at both Joslin and University of Florida. And at each of the sites, we had 6 rats per site per sex time point. Everything else was exactly the same. There's a little bit of phenotype data. So we have pre and post exercise blood lactates. We have body weights. There are some body composition data, although that really is just characterizing the animal, but also things like time on treadmill is also included in this. Okay. And then the design I haven't mentioned much about is the trained response to endurance exercise. These studies were all performed at the University of Iowa. These were done also on 6-month-old Fisher 344 rats and 18-month-old rats, male and female. And like the acute response, the Iowa site collected approximately 19 tissues. But in this case, we're collecting from intermediate stages as well as what we would call a trained state out at 8 weeks of training. Excuse me. The training was included 5 days of treadmill running per week. In this case, Sue Bodine led this arm, and she was able to do VO2 max measures on these animals. So she kept them working at or right around 70% of their VO2 max throughout the training protocol. So that was obviously the workload was increased as they became trained, but it was all very well controlled. All the tissues were collected at exactly 48 hours after the last bout of exercise. The sample size is quite large. It's 12 rats per sex per time point. And there is a lot more phenotype data, and these are just some of the examples of the data that Sue was able to get: body weight, body composition, and VO2 max changes. And like the other studies, the time from death to tissue freeze was recorded and is also available. And the time points we collected after training were after 1 week, 2 weeks, 4 weeks, and then week 8, which was what we considered the trained state. And then really to wrap it up, just Sue Bodine at Iowa, Lori Goodyear at the Joslin, and my group at Florida. And then really a shout out to the team, because you're talking about 19 tissues per animal and hundreds of animals, and it really took more than a village. It took big teams, and they're the ones that made this happen. So thanks to them. Okay.
Okay, thank you, Karen. My name is Nicole. I'm a student at Stanford University, and I'll briefly review the GET assay protocols. GET stands for Genomics, Epigenomics, and Transcriptomics. There are 2 GET sites, Stanford University and the Icahn School of Medicine at Mount Sinai. The external release includes data generated by 2 GET assays, RNA-seq and methylation via reduced representation bisulfite sequencing, or RRBS. You can see in the table on the right the availability of each data type for the 5 tissues in the release and which site generated the data. Notably, both sites processed RNA-seq data from all of the gastrocnemius muscle samples, so there are technical replicates for that tissue as well as biological replicates. I will briefly describe the experimental protocols for these assays. We receive all samples from the biorepository frozen and powderized in the case of tissue. These samples are first homogenized in a lysis buffer, and total RNA is extracted with the Agincourt RNA Advanced Kit, and libraries are built out with Nugent's Universal Plus mRNA-seq kit. Both of these kits facilitate large-scale sample processing, so these steps are automated with biomicrobots, and all samples of a tissue are processed together on a 96-well plate to reduce batch effects. QC metrics indicated in the orange boxes are collected and reported in the release, so you have those values available to you. And finally, after low-depth sequencing for QC and rebalancing of the pooled library, we submit the libraries for deep sequencing with Illumina's NovaSeq. RBS experimental protocol looks similar, except DNA is extracted with the GenCort V3 kit, and libraries are built with Nugent's Ovation RBS Methyl-seq kit. Again, this process is automated on a biomech robot as much as possible, and the recorded QC metrics are indicated in the orange boxes. After deep sequencing, the raw files go through similar pipelines to quantify the results. You can see the particulars of the software used at each step in the table below, but the general workflow is to trim adapters, which includes diversity adapters in RBS library or reads. Then we align those reads to the RN6 rat reference, as well as genomes of known contaminants, including spike-ins. We quantify duplicates, which we're able to do very accurately because of the unique molecular identifiers that are integrated into both types of these libraries. And duplicates are removed in the case of RBS, and quantify results, which are available in the release. Log files at each step are compiled into Pipeline QC report that's also available to you. A big thank you to all members of the GET team that have helped generate these data sets. And now Dr. Charles Brandt from the University of Michigan will introduce the metabolomics workflows.
Okay, there seems to be some audio difficulties from Charles. Josh, would you like to jump on and talk about proteomics first, and then we'll come back to Charles for the metabolomics?
Yes, I could go ahead and do that. Can you hear me?
Yes, we can hear you.
Okay, great. I'm Josh Adkins. I am the Associate Division Director in the Biological Sciences Division and Group Leader of the Integrative Omics Group at Pacific Northwest National Laboratory. And today I'm just going to emphasize the data that is in the public release available to you for this RFA. And so I'm going to, I show in this diagram above the time points that Karen Esser described and discussed. And so one of the points I want to make here is that we used a tandem mass tag labeling technology for this particular experiment that required some constraints in the time points we evaluated. And so I circle in blue the time points that we've looked at. The samples also included 4 proteomics analysis, 6 male and 6 female rats per time point. The Pacific Northwest National Laboratory performed the mass spectrum LCMS methodologies for gastrocnemius and white adipose tissue. The Broad Institute performed the analysis with liver and heart. So this is a diagram of the proteomics multiplexing protocol. Again, we used a tandem mass tag technology in order to perform this study. What you can see here is the workflow where we take proteins, extract them, denature them, reduce them, alkylate them, digest them using a lysine C trypsin methodology. I should say that these methodologies were performed the same way at both the Broad and PNNL. We then aliquot, we then label and then aliquot samples. What we've done in this particular case as well, for each tissue, we've produced what we would refer to as a universal reference, which for each set of TMT plexes or multiplexes, this reference is used so that we can compare across the different plexes. We then take these combined samples, perform a high pH reverse phase fractionation methodology. This creates 24 fractions for global proteomics and 12 fractions for phosphoproteomics. And if acetylation or acetylproteomics is studied, those were 6 fractions. We then used IMAC in order to produce the phosphopeptides. We used Q-exactive class instruments. We then perform data analysis, which produces peptide identifications as well as the quantification information in the same slide. The data is processed by the Broad or PNNL independently prior to being put into the data repository. I hear some feedback out there. So one of the points that I'd like to make for the batching of the TMT11 multiplex design is each TMT multiplex experiment included a male series of exercise time points, which would include the unexercised control immediately post-exercise, 1 hour, 4 hour, and 24 hours. There was a corresponding female series, so that produces 10 individual samples within each multiplex, and then the 11th plex is a common reference. We then randomize those time points and the sex within each multiplexed experiments. So what this produces is, because there are a total of 12 animals, 6 male, 6 female, this produces 6 total multiplexed sample sets or experiments. The pipeline and quantification, PNNL uses MSGF plus and a MASIC data extraction quantification pipeline. The data available includes raw files, and this is gastrocnemius and global and phosphoproteomics and white adipose with global and phosphoproteomics. The Broad uses a Spectra Mill data extraction quantification pipeline. The data available also includes the raw files. Their data production was liver, global proteomics, phosphoproteomics, and acetylproteomics for both liver and heart. The reasons for not including acetylproteomics for adipose is the sample quantity was quite modest for white adipose tissue, and for gastrocnemius, earlier studies showed that the acetylation component of the proteome was very, very modest. So I'd like to just provide thanks to the team members at Pacific Northwest National Laboratory. Proteomics is a fairly labor-intensive methodologies, and so large, important teams on both sides. This is the team from PNNL, and then the team from the Broad. With that, I'm done. I guess turning it over to Charles or Chuck.
All right. So thank you, everyone. Josh, you can hear me, right?
We can
Yes.
hear you.
Yeah, okay, great. I just wanted to make sure. So, first, let me apologize for my technical ineptitude. So I'm Chuck Brandt from the University of Michigan. I'm representing a number of different sites. Can we have the next slide, please? So I'm representing the metabolomics and the lipidomics and the untargeted metabolomics platforms. There are 6 metabolomic sites in the consortium. Targeted assays are being done at Duke University and Mayo Clinic. The targeted assays are, analyzing specific metabolites through high sensitivity and quantitative measures. Lipidomics are done both in a targeted and untargeted manner. Targeted stuff is being done at Georgia Tech, mostly the oxylipins, and untargeted sites are the University of Michigan, where I'm at, and the Broad Institute, run by Clary Klisch. You can see the tissue and the tissues that are available and the data that's available from the metabolomics assays. In the heart, at this point, only untargeted data from the Broad Institute in the positive mode is being done. Next slide, please. So targeted metabolomics, again, is being done at both the Mayo Clinic and Duke University. At Duke University, you can see the members of the team there in the upper left-hand corner of the left box, using various platforms to assay the metabolites that are shown in the slide here. So everything from amino acids to specific pathways such as keto acids and some disease biomarkers. This is done by flow injection, which is a technique. One technique, some of the assays are done by MSMS, which gives you more quantitative measure. I'm going to say at this point that at all sites, the data is initially normalized by in-house protocols that have been worked out and vetted. All those protocols are going to be available to individuals who want to have the details. So at the Mayo Clinic, another set of targeted analysis are being done. There's some overlap between the two. We've been comparing these approaches. Mayo Clinic is being led by Sri Nair and Ian Lanza, and you can see the rest of the team on the upper right-hand corner. Next slide, please. The lipidomics assays, as I mentioned, are measuring most of the fairly non-polar compounds. Emory University is doing the targeted oxylipin assays. These are generated from omega-6 and omega-3 fatty acids. It's done after a solid phase extraction and uses a triple quad to attain the sensitivity that you need to measure these fairly low-level metabolites. And those are listed there. Again, there's additional information available if you need. The lipidomics assays, which is being done, being led by David Gall and Facundo Fernandez, is using also a high sensitivity instrument, the Q Exactive. And these are done in more of the quantification is not absolute, as a relative levels of these different lipids are being assessed. You can see that about 500 lipids are annotated, so you'll have names of specific lipids. And then there's up to 10,000 unannotated lipids in this platform that if you want to dig through that data, it can be done. So this is done in reverse phase and a positive and reverse phase negative mode to get better ionization. The data is collapsed into annotated. The annotated compounds are collapsed such that the lipids with the best measurements in the phase, if there is overlap, is being presented. But again, all the data is there. Next slide. So the untargeted platforms, they really are being run as hybrid platforms at both the Broad and the University of Michigan. And what I mean by hybrid platforms is that a number of identified metabolites, in the case of the Broad, about 200 metabolites are being detected in their HILIC, which is a separation method, and ionization in the positive mode. So they can identify 200 lipids, or excuse me, 200 metabolites with relative quantification. And then thousands of unknown features are present in the output from these from this platform. Similarly, at the University of Michigan, we do reversed phase chromatography prior to mass spectroscopy. It's done both in the positive and negative mode. And we also do a platform called ion pairing, which is also run on the form of reverse phase column, but gets more, a higher preponderance of negatively charged small organic molecules. And so, oops, one slide back. So you can see the number of named compounds is in the parentheses there, and then the class of compounds that you can see. And again, both at the Broad and the University of Michigan, the team members are there. And I'll stop there.
Right. Hi, my name is David Amar. I'm a computational biologist at Stanford University. And I will walk you through the different files that you're going to see when you download these data. And we're going to start with the phenotypic data set. And that includes sample level information, which could be either technical variables or biological covariates for each sample. So that could be the weight or the sex of the animals, for example. These data sets or this information is useful for almost every type of analysis for the omics data sets. And we provide these data as a single large table with a row for each sample ID, which we call the vial labels. And these are linkable to the organism ID, which we call the PID. So the columns in these data sets are different data fields, and they're all explained in an additional file that you will see, which is called the data dictionary. And we provide these data sets either as a simple tabular text files or as R data files that you can load directly into your R session. So these are some notable columns that you will see once you download these data. The first is the vial label. As we mentioned before, this is the primary sample ID. Second one is the PID, which is the animal ID. The third one is a textual description of the randomization group. So this includes both the time point of the rat and whether or not it's a control or an exercise rat. We then have sex, the distance achieved during the exercise test, the weight of the rat, the time to freeze of the sample, and a textual description of the sample, which includes the tissue. Now, this is an example of how the table looks like and how it is useful. So we have here at the top an illustration of these phenotypic data. We have a row for each sample ID. And then when you will go ahead and get the molecular data sets, you will see that we have the same IDs as columns in the different numeric data tables. Now, these IDs could be different between different data sets, even of the same tissue. However, you can use the phenotypic data to map them to the same animal ID, and thereby you could merge the data sets and do integrative analysis. So we'll now move to the transcriptomics data set. Here we have the general structure of the directories that you will see once you download and unzip the data. This structure will be consistent for in most cases for the different omics as well. So we have the first directory has the name of the omics, which in this case is transcriptomics. We then have a directory for each tissue, and we also have a directory for QA/QC results files. In this case, we keep 2 metadata files of the samples in this directory. One is the pipeline QC scores, which is metrics that we compute while processing the reads from each sample. And the second file contains metadata information about the sample preparation. Now, for each tissue, we have the name of the assay. In this case, we have one transcriptomics assay, which is RNA-seq. And then we have the results for that assay in that particular tissue. In this case, for RNA-seq, we have different data tables that provide gene level information for each of the samples. So this is a simple illustration of the file structure and how you can use those. So here we have in the middle the gene count table, which could be used for different tools for analysis of RNA-seq data. We have a row for each gene, and we have the counts for each sample. Now, you can take the sample IDs and obtain sample level information from the metadata files. The first one could give you information about the preparation of the samples, for example, the RIN score. And the second file can give you information about the output of the pipeline that we used to process the reads, for example, just the number of reads, the percent of GCs, and so on. Now, we provide the simple gene count dataset, which most analysis tools for RNA-seq could use, but we also provide the FPKM and TPM matrices, which are normalized counts at the gene level that you can use for other analysis as well. Now, moving to the epigenomics dataset, we have one assay in this external release, which is the RBS methylation data. You could see that the directory structure is similar to that of RNA-seq. So we have the same or similar QA/QC folder and a similar structure of the folders for the actual data. However, the results file are different. The results file for the RBS are Bismarck files. So this means we have a file for each sample. Each Bismarck file provides a data table with a row for each region or for each point in the genome. And for each row, you have the counts of the methylated reads versus the counts of the unmethylated reads. Now, again, these data contains the sample vial labels, which are the sample IDs in the name of the files. And these are mappable to the different metadata files that are similar to that of RNA-seq. We have one file for the sample preparation and one file with metrics that we obtained from the output of the pipeline that was used to process the reads. Now, as a comment, Bismarck files are useful and could be naturally used for analysis. For example, if you use the EdgeRR package, you could, in one command, load all these files into your R session and perform different analysis, such as differential analysis or visualization of the data in that package. We'll move now to metabolomics datasets. As you can see, again, we have the same file structure. However, we have a few differences than what we've seen before. First of all, we have multiple assays per tissue in metabolomics. Also for metabolomics, we have two main directories, one for targeted datasets and one for untargeted datasets. But also the third difference is that we have additional files that are called the MoTrPAC Metabolomics Data Dictionary, which are useful when you map the names of the metabolites across the different assays, and I will illustrate that in an example later on. So these are the names of the assays. These are the names of all the targeted assays and the names of all the untargeted assays. So each one could have a directory in the results directory of each tissue. And this is an example of how the actual files or results files will look like. This is an example for an untargeted data set from plasma using the Helix positive platform. We have 8 files here, 4 representing the results from the named metabolites and 4 for the unnamed metabolites. The reason these are separated are, first of all, the results of the named metabolites were produced using an additional step of identification for known metabolites, but also this part of the data set is much smaller than that of the unnamed metabolites. So if you're only interested in named metabolites, it's much easier to just use these files as they are much smaller. We have here 3 metadata files and 1 results file. The first file contains some textual description of the experiment. The second file here is the metadata of the metabolites. And then we have another file for the metadata of the samples. And then we have the actual numeric results of that metabolomics platform. And here we have, again, our illustration of how these files look like and how they could be used together. At the middle here, we have the actual results file of this metabolomics assay. You can see it's a simple table with a row for each metabolite, and then the abundances for each column, where each column is a sample. Again, we have the different sample IDs in the column names. These are mappable to the metadata file of the samples in this directory, which you can use, for example, to obtain the sample order by which these samples were produced. Now, for each row in this results table, we have another metadata file, which contain information about these compounds and these metabolite names. So you can see we can have different columns in this metadata file. We can have, for example, the retention time of the metabolite and so on. And this file could be different between the different platforms. So when you compare these files, you could see different output for each assay. However, all of these files have these 2 columns, which is the MoTrPAC compound ID and the MoTrPAC compound name. And these are linkable to our MoTrPAC metabolomics data dictionary. So if, for example, you're interested in a specific metabolite and you want to compare its replicability across the different assays, you can use these columns and the data dictionary to map this metabolite and obtain its results from the different data tables. Now, as another side comment, keep in mind that metabolomics targeted data sets usually report the absolute concentrations, whereas metabolomics untargeted data sets report relative intensities from the peak areas that are identified. So that means that the scales of these data sets are not exactly the same, and you can obtain more details about these issues in the experimental details file that you have in each directory. We now will move to proteomics data. Again, we have our directories. This is similar in spirit to metabolomics in that we have a few assays for each tissue. In this case, we have up to 3 assays per tissues, 1 global proteomics, another one for phosphate proteomics, and in some cases, whenever we could produce, we have another folder for acetyl proteomics. This is the general files that you will find in the results of each assay. We have here, and I will focus on those of global proteomics. We have here 4 files, 2 metadata files, 1 for the samples and 1 for the proteins or peptides. And we have 2 results files from the assay. So these results file contain the numerical abundance results from the analysis and the pipeline that processes the raw data. And the first file is the reporter ion intensity. You can think of this as the raw data that we get from the pipeline. Now, as was explained before, these data are partitioned into plexes, which you can think of them as small batches of 11 samples at a time. And you can obtain a mapping from the sample ID to these batches in the vial metadata file. Now, one traditional pre-processing step in proteomics is to take the data from each such plex and compute the log2 ratios between each sample and the reference channel that is present for each of the plexes. So this provides data that are better normalized across the different plexes. However, we provide both of these files as different groups may have different types of analysis that they we prefer to use to normalize the different plexes. And again, we'll go through our illustration of how these files look like. We have here at the bottom the raw data, the RII data set, which is the raw intensities. We have a row for each protein with a few annotation columns, the PTM ID, the protein ID, and the peptide. And then you have a column for each sample with the numeric values as we had in the different data sets before. And then these data are used to compute the ratio data sets. So you have the ratio files that contain, again, a row for each protein and the sample level information. Now, for each protein, you can map the ID of each protein to the ratio ID metadata file, which contains the metadata of the different proteins. And you can use this file, for example, to map the proteins and the rows in the results file, for example, to interest gene IDs. And the last file that we have, the fourth file, is the val metadata file that you can use, as we mentioned before, to map the sample IDs, for example, to the different plexes that were used to generate the data. So after we went through the file structure and the different files that we have when we download the data, we also provide with this webinar a simple R notebook example that you can use as a starting point to see how you could merge the different data sets from the different assays. Now, I will not walk you through the R code, but we have here general guidelines, and you can download this R notebook from this GitHub link that you could get from this presentation. But to run this R notebook, you will first have to download all of the data sets. So we have a link here to download all the data in one directory. In this case, you unzip that directory into your desktop. That's the assumption that this R notebook makes. And then you can follow the code and the commands to run the different steps of this notebook. And just to give you the general steps that this notebook can do, we have here the first steps. You will load the phenotypic data. And for this example, the RNA-seq data and the proteomics data from liver. Then we illustrate how you can use the phenotypic data to merge these data sets. Keep in mind that the proteomics data set has a few less time points than that of the RNA-seq. So there is a step that you will need to go through the animal IDs to merge these data sets. And then we give one example of how you can examine the correlations of a specific gene and obtain plots, for example, as the one shown here. So now I will go back to our file structure and go through the QA/QC part. So for each assay, for each omics type, you have a QA/QC folder. And we've seen before that in some cases, for example, in the RNA-seq, you have useful information there that is the output of the pipeline that was used to generate the data. So these are submitted by the members of the MoTrPAC consortium that produced these data set and ran the pipeline. However, we also have an HTML file here that contains an R notebook with a secondary analysis of all the data sets combined. So this secondary analysis provides a more involved R notebook that you can follow and use its output. And I will just walk you through just the steps that this notebook can perform. They load the data and all the libraries required for the analysis. Then we have a proposed step of data filtering and normalization. These are very simple steps, and more advanced analysis and normalization, you can add them for your analysis. But then these data, once normalized, are used to compute correlations and principal components. And then you can plot, for example, the PCA of each data set and check whether the principal components are correlated with technical or biological variables. We also report flag samples in the RNA, in the PCA plots. So keep in mind, all of the samples that are provided with our external release passed all of our QA/QC steps. However, sometimes when you plot data in the PCA plots, some samples may look a bit different than the others, and we just flag them in the plots. You can use these flagging maybe to perform some sensitivity analysis later on. And also for some omics, we have additional omics-specific QC steps. For example, from sequencing data, you can use the sequencing results to check and make sure that the sex of each sample matches the sequencing data. And with that, I will give the presentation back to Amelie.
Okay, so how do you access data on the MoTrPAC Data Hub? So first, I want to clarify that MoTrPAC consortium members do not need to register for access and not sign the user agreement. So everyone who have access to the internal MoTrPAC.org page do not need to register. And if you're uncertain about access, please contact us at motrpac-helpdesk@lists.stanford.edu. All other users must register to gain access to the data. This involves signing a data use agreement that specifies the terms for data usage, filling in your specific information, and creating a user password. And before we move on to a demonstration of how to access and download the data, I just want to mention a few file characteristics that you can see here on your right. So all data sets are in a compressed format. The data on the hub don't include any raw files. So if you're interested in accessing the raw files, please contact us at the help desk email that will be listed again at the end of this webinar. And for Mac users, you can just double click on the downloaded compressed file to extract the data. For Windows users, we recommend using either WinZip or 7-zip to extract the data. And this is the MoTrPAC Data Hub. I will now walk you through how to access the data. And if you've already registered, you can directly log in through the login button. Otherwise, you press Data Access. This will get you to the external release page where you find study documentation. Here you will fill out the data use agreement that specifies the terms for use of this initial data set. We are asking researchers to agree to a publication embargo that is valid until January of 2021. Here you'll also register your personal information. You will need a personal e-signature, first, last name, email, and affiliation. Then you'll receive these two emails, one's to welcome you, and one where you click the link, you can see here, and set up your password. Please check your spam folders if you do not receive any email. Once you've entered and confirmed your password, go back to the Data Hub and use the login button in the top right corner. You can now use your email and newly created password to log in. This will take you to the release page where you can download the different types of data. You can click to download the specific data that you're interested in, or all of the available omics and phenotypic data at once. Please note that you're downloading large files, so some are several gigabytes. It may take some time to complete. When you press download, this generates a link for you to download the data. And you can see up there how that you get your specific data that you choose to download, in phenotype data in this case. And we provide more detailed information about data access, data structure, and data download in the README that you can see on the top here. So please take a look at the README. Contact us if you are interested in accessing the raw data, or the MoTrPAC help desk if you have any questions. I also want to emphasize that this is a preliminary data release, so more features on the Data Hub portal will be available as the study progresses. All the information presented at this webinar and more is available through the MoTrPAC Data Hub. Here you can find the protocol describing the animal study in detail. the MOPS for Manual of Procedures, where you can find assay-specific information about how the samples have been processed and run. There is one for genomic, epigenomic, and transcriptomics, or GET, one for proteomics, and one for metabolomics. There are QC SOPs for standard operating procedures that describe the quality control workflow for each data type. And I again want to emphasize the README that contain a lot of useful information on how you download and use the data, including, for example, what the different columns mean and how you can connect sample IDs with time points of exercise, for example. There is a link to the R notebook that David just described for your use. You can also find additional information about MoTrPAC at motrpac.org. And again, if you have further questions or comments, please contact us at the MoTrPAC Help Desk email. This webinar recording and these slides will also be available to you through the Data Hub. And with that, I would also like to acknowledge all the members of the bioinformatics team here at Stanford, and especially mention our team of developers, Karen Dalton, Jimmy Sen, and Young Kim, who've done an amazing job building the data portal so we can share the MoTrPAC data resource with you. And now I will hand over to John Williams from the NIH, who will give you some information about the RFA. There seem to be some additional audio problems, but while we get that organized, we also have a slide that summarizes all of the available assays per tissue. So if you're interested in a particular assay or want to know what all the assays for the particular tissue that you're interested in, this slide will also be available. And I also want to remind you that after John has described the RFA, that there are two ways to ask questions. After the webinar, you have the Q&A function, where you can also choose to ask questions anonymously, and the raise hand function, if you want to ask your question orally. And our different speakers and also additional MoTrPAC members are available to address them.
So this slide is just meant to illustrate that there's a funding opportunity now available for additional phase 2 animal studies. Provides the contacts listed below at NIH if you're interested or need to know more specifics about the RFA itself. The letter of intent is due February 20th with the application date due March 20th. So I encourage you to get in contact with John if you have specific questions about the RFA.
And with that, we want to thank all of the speakers today and open up for questions. Please go ahead.
You can just hi, this is Karen at the BIC. We've got some additional questions that have come in. Ashley Jha has commented, Ashley Jha from NIH has commented, data from additional tissues are being prepared and data will be released in the future. And we have someone asking, is today's slide set available for download? This slide set will be available for download, as well as a link to the recording of the webinar that we provided today. Len, do you have anything else for now?
No, I think we will keep it open for questions.
Excuse me, this is Ashley. There's one question.
We have a question about the more information on the RFA. So if anyone from the NIH would like to answer that.
Could they elaborate what the question might be?
We have a question about, for the RFA, do applicants need to determine the candidate molecule to study the mechanism? And then we have a general comment about more information on the RFA, where that can be found.
So I think, so this is Karen Esser. I'm not an NIH official, but as someone that has talked to John, unless he's actually on here, in terms of the specific mechanistic hypotheses question from Anthony, I would suggest you talk to John. Actually, I think my understanding is that they're not looking for single molecule, single pathway grants in this, but rather obviously take advantage of the data set and you know, because there's a lot of data, and the overall goal of MoTrPAC is to develop ideas about some of the molecular transducers. So do you have ways to sort of sift through either assays or screens or informatic tools? But I don't, you know, so I'm just saying this as someone that has talked to John, that's part of, and as part of the RFA. I want to reinforce that the most important person to talk to about this is John. But I know we've had discussions that he wasn't really looking for a single molecule, single pathway kind of grant in response to the RFA. In terms of the from Dr. Tu about the MoTrPAC list, so as Ashley alluded to, the assays, a number of different assays have been done on both phase 1A and phase 1B tissues. For the RFA, it's only the 5 tissues that are publicly available right now, and so just use those. But obviously, at some point in the future, the data from the rest of those tissues will be released. Oh, and so actually, Joe Moore, you asked about control samples. This is actually an active area of discussion. So for the phase 1A samples, we had only included 2 controls, one at 0 hour, one at 7 hour. We are actually in the process, especially for the time course study, to go back and provide some more temporal resolution with also with attention to fasting time as well. So it's not finalized yet. I mean, so nothing's going to be released before the RFA, but we are in discussions right now to add some additional controls for the past studies.
Just to clarify that question from Joe Moore, so that there's 5 male control animals and 5 female control animals at 2 of the time points for RNA-seq data, since you asked specifically.
This is actually to answer Chris Ward's question regarding whether there's a differential express gene information. That is currently actively under analysis, and you know, I think we're not in a position to release that result. So if you are capable of doing that, you may want to do it yourself.
Okay, if there are no further questions, I want to thank all of the speakers.
There are more questions, Marlene. There's a question from Joe Moore. Will FASTQ files be available if we request them?
Yes, I didn't see that, sorry. So yes, FASTQ files will be available. So please email the MoTrPAC email if you want access to FASTQ files.
So the questions regarding the RFA, it's best to direct to John Williams directly.
Unfortunately, John Williams seemed to have some problems with his audio. But of course, his email is in the slide deck here. So if you have specific questions about the RFA, please reach out to him.
Hello?
Hello.
This is Zen Yank from University of Virginia.
Hi, please go ahead.
Isn't an effort to make the data user-friendly? Right now, my problem is that You know, you can go to the data, you can find the ID and find the phenotypic data and try to link them. But if you put a time course together just as a, you know, time course response to exercise for any one of the parameters, do you have an effort to do that so that everybody can look at it quickly, say, okay, there is a quick response of muscle to, you know, running with PGC1 upregulation, something like that, and then try to find, you know, it's just a user-friendly. I'm not a, you know, omics person, so it's very difficult for me to do that.
So
This is Karen at the BIC, and this is a preliminary data release of the earliest sets of tissues to support the RFA on an ongoing basis. There will be multiple ways in which the data for future releases will be presented in a more integrated fashion. This was a way for lots of different labs to submit data to us for us to provide it in a cohesive way to support this RFA. So in this preliminary version, this is the version that is available to support the RFA. And that's what we have at this time. Going forward, of course, it will be in different formats.
Yeah, this is Ashley from NIH. I think the suggestion is very, we heard your suggestion, and I think this is a very critical one. In the future release, we probably would like to do something, make it more, you know, post initial analysis kind of a result. However, this result was released hot from the press. That's why we have embargo period post on it, because the consortium haven't been able to look at the data closely yet. So we heard your suggestion. This is a very good suggestion that in the future we'll definitely will consider release in a different format that's more, you know, intended for biologists.
And I think I'm just looking at Anthony's question here, and anything related to the RFA, ultimately, you really should, I mean, you just talk to John Williams. I've been exchanging a few emails with him. He really is very apologetic that for whatever reason the audio didn't work, but he's more than available to answer questions. So please contact him. And in terms of, I have no idea, but I don't, in terms of whether we'd be reviewers, I don't know. I don't think so, but I have no idea.
Okay, if there are no further questions, so if you have additional questions about the data access, please contact the help desk email. If you have specific questions about the RFA and what the content of that, please contact John Williams at the NIH. And with that, I would like to thank all of the attendees for joining us today and all of the speakers. Thank you very much.